So my sister raises alpaca, and when I visit, sometimes they do spit on me. Do I need to be concerned?
Hello, everyone, and welcome back to People, Parasites, and Plagues, a podcast aimed at delivering information about the fascinating pathogens among us from the scientists who study them. I am Kim Klonowski.
And I am David Peterson, your host for today's episode. Can a fast immune response beat a slow replicating pathogen? Don't bet on it.
Our guest today is Dr. Fred Quinn, head of the Department of Infectious Diseases. Fred, welcome to the podcast.
Thank you very much. It's a pleasure.
So what we like to do is start out with a question about your career path to scientist. You can start in high school or kindergarten, wherever.
Oh, boy, that's a tough one. Well, I started as a newspaper boy delivering papers, but no, I started thinking about infectious diseases really not until I got into graduate school. So I actually wanted to start my career looking at microbes that would eliminate problematic trace metals on the planet, like mercury and cadmium and things of that nature. And tell me if I can talk about this or not, but it was a certain political era. And my boss told me that probably the president at the time wouldn't support this kind of research. So maybe take your trace metal studies into infectious diseases. So I did, and started looking at iron acquisition by pathogens like Legionnaire's disease, Legionella pneumophila, and then ultimately got into host pathogen interactions in my postdoc at Stanford. And then I was introduced to the CDC by my co-mentor, Stanley Falco, at Stanford, who said at the time it was a good idea to go into the CDC because NIH budget got cut. And so you may not want to go to academic research.
So a strategic move.
It was a strategic move. And I said, okay. I interviewed, and I spent 13 years at the CDC, and it was wonderful. I had a really, really good time in bacterial emerging infectious diseases. During the HIV era, tuberculosis became the number one emerging bacterial disease that I worked on. And then towards the end of my time there, I got to know the College of Veterinary Medicine here at UGA because the high containment laboratory here was getting built, and animal modeling was an area at the time. CDC has very good animal modeling now, but at the time it was a little thin. But here it definitely was a concept that would be very, very well received in the field. So moved here and really began developing studies, not just in animal models, but in human cell lines, as well as bacterial genetics, etc. And then more recently, in the last 5 or 6 years, we have gotten very much involved in looking at international field sites, because most tuberculosis is international, and a very large part of it we're finding is because of animal to human transmission.
Oh, wow. You've given us a lot to talk about today.
Definitely.
So maybe we should start, because I don't know if we've asked anyone this before. What was it like to work at the CDC?
It was great. Again, it was a very important area to help assess the basic research. It wasn't an area of huge amounts of basic research. It was mostly emergency response, at least our area was. So if there was an outbreak of some disease, there are investigators, EITS officers that go there, and then we work with those individuals. They send us samples, we do processing and analysis. And so you tend to hop from one crisis to another. And it's very exciting and very fulfilling. But there is also a little bit of a side of it where you have ideas about, maybe a diagnostic here or a vaccine here, and you have to kind of set it aside because the next crisis has...
So is that one of the reasons why you wanted to come back to academia?
It was.
Yeah.
Yeah.
And was that hard to get back to come back into academia?
Well, I mean, again, I really enjoyed my time there. I really enjoyed the people I worked with, and shifting pretty much to a full-time effort of basic research as opposed to response when something is going. That is kind of now being fulfilled because of our work in the developing world. Looking at these transmission sites and animal, trying to understand transmission from animals to humans, etc. So it's, I think now we're kind of looking at both sides of the equation again, which is great.
So I know that you got a lot of projects going on in your lab, but if you could narrow it down to a fundamental question that drives the research, or we'll even let you have two questions if you need it.
That's a good point. I would say I probably can narrow it down to one, and it's we're looking at transmission. So we're trying to understand it from an epidemiological standpoint. We're working very closely with Chris Whalen and his group in School of Public Health here, and with our counterparts at Makerere University in Kampala, Uganda. And with that group, trying to understand how the organism transmits. You would think it's been in the human population for 12,000 years. You would think we would understand how it transmits. And, the common point is, well, somebody coughs and you inhale it like COVID. It's a lot more complicated than that. And trying to understand this has been the work of a lot of people, including Dr. Whalen. Just to give you an idea, very intensive work he does where he's looking at all these different facets of transmission, doing contact tracing, we do the whole genome sequencing and comparative analysis. Even with all of that, even using cell phones to try and look at transmission patterns with permission.
We want that to be very clear.
Very clear, very, very clear. But even with all of that, we can only identify 25% of the sources of the transmission.
Oh, wow.
So there's a lot about this organism we don't understand. And that's an area we're getting into with the carriage state now. So
I was just going to ask about, studying transmission. So I imagine there's not really a good animal model for TB, right?
Well, there are good animal models, but they're expensive and they're complicated to work with. Obviously, the mouse model is the most used. The problem is the mouse does not cough or sneeze.
Right.
It doesn't transmit. So you're looking at an end phase because you're putting the organism in the lungs and you're looking at the end stage of the disease. There are better models, more expensive and more complicated, and the immunological reagents aren't as good. So, for example, the guinea pig is the next most widely used model, but which it does a little bit of transmission from animal to animal, but it's not great. But it is actually being used very interestingly to look at human to guinea pig transmission in a study in South Africa. And this is very interesting because unlike the mouse model where you infect it and all of the mice get sick and have full-bore tuberculosis, when you do it this way, it looks more like the human population, where some people get sick, some people don't, some people clear it. And when you do this kind of a natural transmission instead of, what was the word I'm looking for, the actual laboratory infection, when you do it naturally, it looks more like what happens in the world. And so this study is actually quite effective. And then as you work your way up the ladder, the next model that we've been pushing for the last few years is the ferret, because it's very well used in the influenza world. And we've demonstrated that it also you can infect it with the tuberculosis bacteria. And actually it's in the possum family. And the possum is a huge contributor to tuberculosis, cattle tuberculosis, in Great Britain and in other places. And
who knew?
I know, right?
Well, I'm sorry. Yeah.
Fred did, but
let me take a step back. The possum is the transmitter in New Zealand to cattle. The badger is the transmitter, and it's also in the ferret family in cattle in Great Britain.
There are multiple things there I did not know.
No, I know. I'm thinking I'm learning something new. Many things new today.
So I guess next thing I wanted to ask, taking an overhead view of TB research, what are the biggest challenges?
Oh, there's lots of challenges. So one is, I would say, is again our understanding of the transmission process. As an example, the vaccine that was developed in 1920, the BCG vaccine, it's the most widely used vaccine in the world, and it does have protective efficacy because it's given to children at infancy. And it is fairly protective against children for 7 or 8 years against not only pulmonary tuberculosis, but, and this is what isn't discussed very much, is tuberculosis is actually also an extrapulmonary disease. So it affects a lot of other organs. The second most common form of tuberculosis after pulmonary is actually meningitis,
Okay.
and it's highly fatal rates in children in the developing world. And so the BCG vaccine is actually quite protective in children against TB meningitis. So it will always be used until we get this disease under control, but it's not effective in adults and the elderly, the older adults. So there has been a push for the last many years, decades, to try to find not a replacement for BCG, but a booster that will then be protective for adults, because just giving BCG to adults has not been shown to be very effective for some reason. So these extra vaccines have been the NIH has been funding this, the Gates Foundation, lots of places are looking. There's a number of them in clinical trials around the world. Maybe we'll get lucky. We have 3 of them here that we're actually investigating. And so again, a lot of the problem is we don't understand the transmission process. So the vaccines we're using may not be protective because of our lack of knowledge of that stage of the disease.
Well, I think that's an important point, too, is that you need to understand how people are infected because you need to understand what part of their immune system is responding
Exactly.
so that you can target or improve that. And I guess we don't talk about that as much, right? So some people are of the opinion that if you have a respiratory, organism, that you really should have a respiratory-based vaccine.
Right. And
But
BCG is not a respiratory-based vaccine.
nor is things like the flu vaccine. So it is an interesting discussion.
Yeah. Yeah, and again, an area that is not discussed very much is Louis Pasteur invented pasteurization in the 1800s for tuberculosis because it's an ingestion. You get it from contaminated milk and cheese from infected cattle and other animals, and you can also get it from humans through close contact through that pathway. So that's not, again, also very well understood. How does it get from the gut to the lungs?
Mm-hmm.
So this is a lot we need to study more.
So another thing, and you alluded to this earlier when you were talking about the era where HIV first broke into the human population and how TB really blossomed at that time. So that clearly shows that co-infections are kind of important. And is that true just for HIV, or is there interactions with other co-infecting pathogens?
That's a really great question. And I can answer that in 2 ways. There's a lot of work being done now on comorbidities for tuberculosis. These aren't, co-infections, but things like diabetes and other facets of comorbidities are very, very rampant, particularly in the developing world. But co-infections, yes, like HIV, has been demonstrated to be very prevalent in the tuberculosis community. We're actually investigating whether influenza co-infection is an initiator of tuberculosis, because the highest rate of tuberculosis in the world in the 1900s was, guess what? 1919.
So just after the 1918 flu epidemic.
You got it.
Yeah.
Wow.
And so we're trying to understand, we're doing the ferret study to look at co-infection. And there was we got a President's Award to do that, which was really nice.
Oh, nice.
And we haven't quite finished it yet, but that last phase of the study is ongoing. But there's also a lot of hypotheses about co-infections in the developing world, such as parasitic infections. So we have a study that we're going to try to do looking at schistosomiasis
Oh, yeah.
co-infections, because if you look very carefully, those are very, very prominent, those kinds of infections in tuberculosis endemic settings. So it could be altering the immune response to make you more susceptible.
And most of those parasitic infections would not involve the lung directly, so it's a more systemic immunological
Exactly.
disturbance.
You got it. That's exactly right. Yeah. A faculty member who used to be here, Vanessa Zenwa, did a study in South Africa in water buffalo, where she demonstrated that water buffalo, which got tuberculosis, also had very high levels of parasitic infections. And when she would treat these water buffalo with anti-parasitic drugs, thinking, okay, we could switch the immune response so it's now be a lot more effective, she actually saw the reverse, that these cattle actually had, the herds actually got more tuberculosis. And the bottom line is you have to treat both diseases. So because all you're doing is then extending the life of these cattle to infect other cattle.
And this is why we do science.
Exactly.
That was a surprise for her to see those data.
So we've talked about the fact that TB can have different forms, which I'm sure we'll get back to again, right? Respiratory, not respiratory. But we haven't really touched on the fact that, TB can be, an active or an acute infection, but it also can be a latent infection. And I'm not sure that our audience necessarily knows what that means. Can you talk about latency and maybe what causes that and if it's a win for the immune system?
Yeah. So it's been studied for many years because it was recognized basically when if you remember which is still given, the PPD skin test.
Mm-hmm.
And it was noted that many, many people tested positive for that, but there was no symptomology for the disease.
Right. And we should remind our listeners that the PPD is the skin prick test that
Yes.
people have gotten to test to see if
Correct.
the test to see if you've ever had TB.
It's supposed to be a diagnosis,
Right.
And so the problem with that test, it's immunologically based. So all it tells you is you've had some kind of exposure. But there's also another facet to that test, and why it's not as widely used anymore, because it also cross-reacts with other species. There's 195 species of Mycobacterium, and some of them will, if you're exposed long enough, you will cross-react with that test. And so there's problems with that.
Yeah, you
So
be careful you don't get put in an isolation ward for not much.
yeah, so there's a new test out called the interferon gamma release assay, which is a blood-based test, and it looks specifically at Mycobacterium tuberculosis gene products that then induce gamma interferon in your cells, which is a better indication of very much the species that cause tuberculosis. There's actually 12 species that cause tuberculosis too, but all of those are in that group. And then so that is one real positive thing about the IGRA that they call interferon gamma release assay. But anyway, getting back to the point we were making, that test still, the IGRA or the skin test, basically only tell you if your immune response has been activated against the organism. It doesn't tell you what kind of disease, quote unquote, that you have. So active disease is the one that we recognize most. That's where you have a horrible cough, you're losing weight, have a fever, night sweats, the whole bit. It's very, very characteristic. It's been that way for thousands of years. But the majority of people who test positive for the skin test or the IGRA don't have any symptoms. So what they have been calling those people for decades are latent. And if you do some research, there are a number of individuals who are latent, who have what are called lung granulomas. So these are structures that you see on x-rays. So usually if you test positive, you have no symptoms, but you test positive, the next thing is a physician will have you get a chest x-ray. And what they're looking for are these structures called granulomas. These are basically containing Mycobacterium tuberculosis bacteria, but it's walled off by your immune system. And basically you could live with these the rest of your life.
So in that case, it is a win for the immune system.
It is a win for the immune system unless you have some kind of an activity that causes problems with your immune system, such as HIV.
Right.
So this is why tuberculosis exploded during the HIV pandemic, because all these latent cases reactivated into active disease.
Right, because HIV was targeting the part of the immune system that was suppressing the active disease.
Yeah, it was building, because the granuloma structures in your lungs are actively continuing to form. And so the HIV virus attacks some of the cells that make the granuloma, and they collapse.
So the granuloma is not an iron cage that encases the bacteria. It's just like an open field with a bunch of guards around.
I was just going to say, it's a bunch of T cells holding hands around the bacteria.
Well, there's macrophages and T cells in there too. Yeah.
There too, and then, HIV
But it
breaks the chain.
It breaks the chain. But yeah, and that's a really good point. And so if you read a bottle of, say, steroids that you have to take for other diseases, on the bottle it says may cause tuberculosis. The steroids don't cause tuberculosis, but it could reactivate a latent infection. So these are, again, areas that are well studied. But you could also test positive for the skin test or the IGRA and not have a latent infection. And you can be culture positive in your nose or in your upper respiratory tract and also not test positive, that we're finding out. So now there are 2 other stages of disease that have been added. So these other 2 stages are the parts that we are extremely interested in, because if you are culture positive, but you don't test positive for any of the current tests, guess what? You're transmitting.
I would say you're typhoid Mary.
You got it.
And not typhoid.
And that shows that
TV Mary.
sometimes science is 2 steps forward, which causes 1 step back.
Mm-hmm. You got it. So that is sort of our new study. We are with Chris Whalen looking at these, and we're calling them carriers. So it's been known for decades that, like streptococcus, you carry it, but you don't have any disease, you don't have any immune response, but you can transmit it to each other. Why wouldn't tuberculosis be the same category? So that's the study we're theoretically going to get started.
I mean, it's good that some of these microbes kind of keep us in business in some ways. Sadly,
but very true. Yeah. So there's a lot more stages to this, and there may be even more stages, but hopefully we're getting a handle on it.
Now, the numbers I read for latency worldwide were between 1/4 and 1/3 of people on the planet are latent.
That's because of the number of people who test positive for the skin test.
And that the numbers for the U.S. were somewhere around 13 million.
Correct.
Which, that's a lot of people.
It is.
So, yeah. How many of those people have no idea that they're latent?
Probably in the entire world, probably quite a few don't know that, because they don't really get a skin test. In the United States, most people do, because at some point, if you are coming from a foreign country, for example, you pretty much have to get some kind of a skin test, or now it's the IGRA test, just so you can live, which is good, to have that kind of information. But just knowing that, again, is not particularly helpful from the big picture standpoint. So until we kind of get a better handle on these other stages of the disease, just being a skin test or IGRA positive, the CDC recommends if you are skin test or IGRA positive, you go on antibiotics.
Okay.
Yeah.
My daughter had to do that.
Yeah. So and again, tuberculosis treatment is 6 to 9 months. In case of latent infection, it's just 1 antibiotic for 6 months. but that's still, so it's better to know what you've got and not have to take an antibiotic.
So there's a lot going on in the news. And one thing I may have read about is a outbreak of TB in Kansas.
Yes.
So can we talk about this?
Sure. Yeah. So it's these occur. There was actually an outbreak in Atlanta in 2016, similarly. And it's usually in, again, poor areas of town, sometime homeless shelters and that sort of thing, because, again, healthcare in these particular areas is not at the level it needs to be. And so if you look at tuberculosis cases in the United States, and there are still, we're very proud because it's under 10,000 for the first time in recorded history of tuberculosis in the United States, which is fantastic. But it's still 8,000, I think 8,500 cases was the count that I think I saw. That's still a lot.
That is a lot.
And again, these kinds of cases are mostly in poor folks who can't get the kind of health care that they need. And what's positive from our perspective about that is when they are identified and diagnosed, the U.S. Public Health Service uses a method for treating them that is approved by the World Health Organization. But basically, you have to take your drugs every day for 6 to 9 months. And the Public Health Service goes and makes sure you take your drugs every day. So that somehow they visually monitor drug taking. And that is a huge plus. And a lot of other countries around the world utilize the same methodology. And that is extremely effective, because if you stop taking your drugs because you feel a whole lot better, the problem is you can develop drug resistance. And that is a disaster.
That's the bad thing. And I imagine it's really important, especially in situations when you talked about if it's a homeless person, right?
Exactly.
You need to make sure that,
Exactly, because you could reactivate and precisely, and it's drug resistant.
Right. And if it's not being watched or managed in some way. So how do we survey for TB among the population? Is that all reported through public health agencies?
It's all reported through, yeah. So basically, again, if you are working in an area like we do here, if you're doing animal research in tuberculosis, we get screened every year for these tests. If you are new to this country, you get screened. But again, because the case rate is pretty low, very low, most people don't get screened. And so if you are in contact with someone who has been positive, who is positive or has an infection, you should get screened.
And is that contact tracing done by state places, or is it done by the CDC?
It's done by so the CDC gets the data, but all of the contact tracing is done by state, territorial, and other local, like the big city, like New York or Atlanta, or they have their own public health tracing service. And it's not just for TB, it's for, pretty much all
And
of
then
it.
the last question is, you've already commented that TB is global. How do we interconnect there and communicate the surveillance data?
Yeah. So the CDC works very closely with the WHO, and therefore those data, along with the public health agencies from numbers of countries, all work very closely with the WHO to intercalate all of these data. So you can go to the CDC website, you will see data not just for tuberculosis but for many diseases on their website. But then the WHO has a worldwide database, and the CDC's numbers are implanted in the WHO database.
For now.
Don't
want to get too political.
Yeah, we're certainly hoping that because again, I worked over the years and still do, and now with the zoonotic TB part, we're the animal to human transmission that we're trying to demonstrate which has not been looked at very effectively for many, really forever. If we're going to control this disease, we need to know all facets of transmission, and contribution of animal tuberculosis is a big part of it. And so we need involvement from not just the WHO, but the OIE, which is their agriculture arm, and we do work with them as well. So it has to be a team effort. That's the only way we're going to make this work.
And I seem to recall that at one point you were working with camels.
Yes.
So camels get TB.
Yes.
What about other camelids like llamas or alpacas?
Yes. Yeah.
And so that's in the New World?
Yeah. So cattle are the number one animal because it is, they are so prominent and they're unfortunately in contact with humans. And those in areas where cattle are not as prominent in the Middle East and other locations, dromedary camels are actually used for milk and cheese and meat. And so there are, lots of herds, and Saudi Arabia is a great example of, they've got millions of dromedary camels there. And so the tuberculosis rate there, and again, Israel and the West Bank is another example, those rates are actually as high as they are in cattle in endemic settings. But no one has ever looked at the process of infection through a dromedary camel. And so our facility here at UGA, the ARC, we could do that study.
So my sister raises alpaca, and when I visit, sometimes they do spit on me. Do I need to be concerned?
Face shield.
So
You can't be a rancher and wear a face shield.
You can bedazzle it.
well, like humans, you could skin test any animal for tuberculosis.
Okay.
And so
If I see them sneezing or coughing, I'll
you
suggest.
could get a skin test.
Okay.
Absolutely.
So Fred, one other thing, you mentioned that there's 150 plus species of Mycobacterium, and there's one that I know you worked on some starting at the CDC, I think, and you still do or would like to, and that's Mycobacterium ulcerans. So tell us why that's dangerous and interesting at the same time.
So it causes a disease called Buruli ulcer. And it was identified about 100 years ago in Uganda. And the pathology basically is this organism, very interestingly, has a very different temperature range than Mycobacterium tuberculosis, because that Mycobacterium tuberculosis and the other species that cause tuberculosis live at 37 degrees, which is, our body temperature. Mycobacterium ulcerans is a cooler organism. And so it can replicate. It has a lot of the genes in tuberculosis, but it only grows in your skin because of the temperature restriction. But it produces it has a dermonecrotic toxin that it's expressing. And that toxin produces enormous ulcers. And unfortunately, because again, it likes to grow, or it is prominent in areas of the world that unfortunately are very poor. It's in the tropics, so it's usually around the equator, and it grows in stagnant water and other kinds of areas. And so if you get a scratch on your skin and you get this organism, it gets into the adipose cells, starts to produce this toxin, and once the toxin starts to form, antibiotics are not effective.
That is truly horrific.
Yes.
Yeah, just when I think we've heard the worst,
Yeah.
it's worse.
And unfortunately, if I may say, because of global warming, the case rates are actually going up. And so we were actually approached by NIH about a year ago to ask because when I was at the CDC, we were working on a diagnostic test for Buruli ulcer that would identify it before the lesion formed, because then you could treat with antibiotics. But unfortunately, the HIV rates were skyrocketing at the time, and so WHO and CDC had to pull their funds into other areas. So I'm not blaming anybody, but it was pretty hard to deal with that. But now we want to resurrect that program. And I was just at a meeting in Melbourne, Australia, because believe it or not, besides equatorial part of this planet that has this, Melbourne, Australia also has cases because it's a tropical region. So isn't this a little bit shocking the way our environment works? But they have cases there, and they have discovered an animal finally that seems to be carrying this organism, the possum.
Once again.
Yes. And what's interesting is they're finding it in the gut of the possum, and it's not causing any disease in the gut.
Do possums have a lower body temp?
Not really.
Okay, who knows how
Yeah.
how it's
But
surprised.
so we're asking these questions, and we've developed a model here called the naked mole rat, which has no skin, and you can adjust its skin temperature. So preliminary studies, we see human-like ulcers.
When you say you can adjust its temperature, are we talking about putting little sweaters on?
No, actually, it's the other way around. You put them in a room and you lower the temperature a little bit
Okay.
down to human skin temperature.
Then he has on a little swim suit.
Yes.
Anyway.
And they form human-like ulcers. And so what we're going to hopefully do is test our diagnostic on these animals. And then we also want to understand what this new discovery about the organism, because no one's looked at humans to see if it's in their guts. So that study is just starting too in Australia to look at so there's a lot to this organism. Again, it's been around for a century, but it's because of the part of the world it's in, it just hasn't been studied as much as it needs to be. And it also might be mosquito.
Even better.
Mm-hmm. So there's a lot of what's going on that we're trying to discover.
I looked up the WHO data on Buruli ulcer, and for many countries, it just said no data. Is that because they don't have cases or they don't survey for it?
It's a little of both. So it's a disease that is equatorial. And unfortunately, most of the countries on the equator are very, very poor. And so if you have a skin disease, usually they don't go to the extent to diagnose it. They try their best to treat it. And unfortunately, in a lot of cases, it's amputation. So but so many other diseases, affect the skin, but this is a big one. And so potentially, we'd love to get a vaccine, potentially a lot of aspects to this. So we're investigating.
Okay, well, our guest today has been Dr. Fred Quinn, a faculty member in the Department of Infectious Diseases. Fred, thanks for being with us.
It has been a pleasure. Thank you. Really appreciate my friends.
So links to additional information about Dr. Quinn's research can be found on our Instagram account, which is at PPP Podcast UGA.
As always, we appreciate you tuning in today. And if this is your first time listening to the People, Parasites, and Plagues podcast, we encourage you to check out the other exciting episodes launched last season. Please rate the podcast, and if you have any questions or suggestions for future episodes, reach out to us via email at ppp@uga.edu. This podcast is brought to you by the Faculty of Infectious Diseases and the Grady College of Journalism at the University of Georgia. It is supported by the University of Georgia through the Office of Research, the UGA Graduate School, and the College of Veterinary Medicine. Thanks to the New Media Institute at Grady for use of their Studio Not Found podcast facilities, and a special thanks to our production assistant, Sid Wigand, for research, editing, scheduling, and keeping us on track.